All right, we left off on Wednesday talking about creatine phosphate. Okay, talking about creatine phosphate. Our simplest, fastest pathway to make energy. Okay, it works very simply. What I want you guys to do to illustrate this is squish them by each other. I know you're here to like stand up and kind of squish into groups of threes. Okay, stand up, squish into groups of threes. Let's go. This would have been better if I brought donuts for everybody. Okay.
Okay, they could be double, so now we're.
Cool. All right. Now, what I want you guys to do is, I want the person on y'all's left to pick up a pen or a pencil. Okay. Now, here's what's happening. Okay. Person on the left holding the pencil. You are a molecule of creatine phosphate. Okay. You are officially creatine. N is phosphate. The person in the middle, you get to be an enzyme. And you are, okay, you're an enzyme called creatine kinase. Okay, creatine kinase. So what's going to happen, okay, is, and then the person on the right, y'all's right, you are a molecule of ADP. So the way this reaction works, okay, is, let me do this, I got an idea, okay. Person on the right, pick up a pencil also, pick up a pencil as well, okay. You're no longer a molecule of ADP, you're gonna start as a molecule of ATP. And you are the definition of energy, okay? So, what I want you guys to do, okay, what I want you guys to do, person on the right, ATP molecule, I want you to set your phosphate molecule, set your pencil down on the, wait, just set your pencil down on the desk, okay, do it, boom. You donated the phosphate to the Coffbridge cycle, to the myosin ATPase. Now you are ADP. What I want you to do is gently tap the enzyme on the shoulder. Good. Now, enzyme, I want you to grab the phosphate from the creatine phosphate. Don't be stingy here. Now, hand it to the ATP. Okay? Now, you've gone from being ATP, ATP, and you're back to ATP. Now, what would happen in regular circumstances now is ATP, put the pin back down on the, there you go. You now donated another molecule of phosphate to the myosin ATPase, or the calcium ATPase, or the sodium potassium. And so, what would then happen is there'd be a line of creatine phosphate molecules behind the person on the left. And okay, I'm going to do it again. Tap the enzyme, grab it. We just go that way until the line is completely depleted. Okay? Does that make sense, guys? Okay, go ahead and sit there at the time.
What happens to creatine phosphate after it donates its phosphate? You didn't specify, I was just.
Oh, we're gonna get to that.
Okay.
Just a second.
Okay.
Okay. We're gonna do the same exercise in a minute, glycolysis, and you're gonna see how it's like playing the telephone game. You guys remember the telephone game from when you were little? Okay. And there's gonna be like 13 steps, and so it's pass, right? And so it takes long. So the reason this pathway is so fast is it's one simple step, okay? We just did an example of this. Creatine phosphate, creatine kinase, right? ADP molecule. And so when the ADP stimulates the creatine kinase, taps it on the shoulder, then creatine kinase pulls the phosphate, and the creatine puts it onto the ADP and we get ADP. Okay? That happens faster than you can even think about. And then it happens over and over and over and over again. Okay? There's a finite amount of this stuff, right? You can imagine that there's like only as much creatine phosphate as there are like people in one row or something. So when they're gone, if I still need ATP, I gotta get it from someplace else. Okay? I gotta get it from someplace else. I raised an important question. We'll point out this in a little bit more detail later. But after you gave up your pencil by creatine phosphate molecules, you're just sad molecules of creatine floating around in a muscle cell, right? You're like, there, what am I doing? I don't know what's happening. Well, when we stop exercising, right? When we stop exercising, you're doing things, we stop. We run the reaction in reverse, okay? There's a creatine kinase molecule that sits in the membrane of mitochondria. And as the mitochondria make ATP, they just grab a phosphate, hand it to the creatine that floats by, and we make our creatine phosphate back. Okay? That's how that goes. So, I jog in place, boom, boom, boom. You tell me my creatine phosphate, and I stop. I immediately begin resynthesizing creatine phosphate. Immediately. Okay? If you deplete all of it, it has a half-life of coming back of somewhere between maybe 30 seconds to a minute. Comes back pretty fast. If you wait approximately 2 minutes, you have about 90% most of the time back. You guys want to know how we arrived at the rest period? They're typically given between sets of resistance training. It's based upon course of creatine phosphate recovery. Okay, so 2 minutes, 3 minutes, 5 minutes, right? When you really want to try to get that one rep, and it's like wait 5 minutes, so you can really check your phone and watch some good TikTok videos and those things. Don't tell people you're wasting time. You're like, no, I'm resynthesizing my creatine phosphate. Okay, I gotta get ready for this next rep. But that's why those things are the way they are. People that are better aerobically trained, they have more mitochondria, they have better mitochondria, they recover creatine phosphate faster after they stop exercising. Okay? This is an illustration of that recovery. Here's the front side that we've already done, creatine kinase. Okay, we're doing this. Okay? Sorry, this is the mitochondrial side. Here's the side that we did. Creatine phosphate, PCR, creatine kinase, ADP, ATP. Creatine comes back to creatine kinase in the mitochondrial membrane. Mitochondria makes ATP. It then uses the creatine kinase to put ATP back onto creatine to make creatine phosphate again. That's what's shown in these graphs. This is 100% creatine phosphate stores in a muscle. You exercise and you deplete them, and then you rest, and you can watch the time course of their resynthesis over time here. Okay? These are data from something called magnetic resonance spectroscopy. You put a person in an MRI machine, and you can track the phosphates. So you have them exercised in a magnet, and you can do some really powerful stuff like all of this. Okay? You're not responsible for drawing these graphs, but you can see how there's this really quick recovery, and then you get halfway and then halfway. Usually that's about a couple of minutes. You're kind of way up here, and that's why we typically recommend at least a 2-minute rest period. This sets. Okay. Okay. One other oxygen pathway that I want us to talk about. It's relatively rare, and we don't have a great capacity to generate energy from it, but it's called the myokinase reaction. Myokinase. So when we get really desperate for ATP, we do what we just did, but rather than using creatine phosphate, we use 2 molecules of ADP. And we use an enzyme, shockingly called myokinase, okay? And we take a phosphate off of an ADP molecule, and hook it on to another ADP molecule to make ATP. So in the myokinase reaction, we start with two ADP, we end up with one AMP, so adenosine monophosphate, and a molecule of ATP, like so. Okay? That's how we end up there. We have a limited capacity with this reaction because as we start to use up our ADP, then we lost ADP that we could then rephosphorylate to make ATP. So this happens a little bit all the time, and it kind of maxes itself out if you're doing very, very, very high intensity exercise. Okay? I have a slide in a minute that shows the kind of relative scores of energy in these phosphagen systems. And you'll see myokinase, we have a relatively small amount that's there, but it's still a significant contributor if we need it. Fernanda. So ATP has two phosphates. So you have an adenosine molecule, right? And if it has one phosphate hooked on, it's called adenosine monophosphate. If it has two phosphates on it, it's adenosine diphosphate. And if it has three, it's adenosine triphosphate. Okay, it's a good question. I know that the biochemistry of all of this is tricky, and so I appreciate the question. So all the things that we do are about transferring these phosphates. I think I mentioned it the other day, but the enzyme that says kinase enzyme means what its job to do is to move a phosphate or to put a phosphate onto something. Okay, it's one of our more important kind of intracellular signals is that we will do phosphates. Okay. Okay. Again, this is just for a relative view for you guys. You're not responsible to memorize these numbers or anything else. But this is a table trying to show the phosphagen system stores in a typical average person's skeletal muscle. Okay. And so you can see we have, this is in kCals for total muscle mass. Don't worry about what the units are or what that functionally means. But we're at about 2 ATP, we're at about 8.5 for creatine phosphate. So about 4 times as much energy stored, maybe a little bit more as creatine phosphate. Myokinase is about 1. So there's less ability to even do myokinase than we have stored ATP. It's not very much. And so our total phosphagen system adds up to about 11 somethings, okay? But the vast majority of it is in creatine phosphate, okay? So I just want you guys to understand, very little ATP, more creatine phosphate, but a limited amount, right? And so these two things can get used up, especially during high intensity exercise where ATP demand is high, very, very quickly. Very quickly. Okay. We're going to move on to our next energy pathway. Okay. We talked about phosphagen, limited stores, very fast. Limited stores, very fast, one-step reaction. Glycolysis has a much larger capacity to generate energy, okay? But it is a longer multi-step pathway, so it's slower, all right? And again, I will remind you all that even as you're sitting there, even as you're using up your creatine phosphate, glycolysis is always happening, always happening. Okay. Glycolysis we define as a series of reactions where the oxidation of glucose to lactic acid, okay, is coupled with the synthesis of ATP from ADP. So it's the breakdown of glucose. So when I ask on things what we have, the substrate for glycolysis is glucose. Okay, substrate is glucose. Where we get that glucose is going to matter, and I'll point that out in a minute. Okay, when we start with glucose, that's what goes in. It's very important to understand, and I guess I should have pointed out, All of the phosphagen pathways occur in the cytoplasm. Okay, in the cytoplasm of your muscle cell. Glycolysis also occurs in the cytoplasm of a muscle cell. It occurs in other cells too, for our purposes, in a muscle cell. Now, and then we get more energy, okay? For every molecule of glucose that we send all the way through glycolysis, and we're assuming that this is going to go to lactic acid, which it does not always do. We are going to get either 2 or 3 molecules of ATP for every molecule of glucose. Okay? 2 or 3 for every molecule of glucose. Where the glucose comes from matters. Okay? Because, and the reason that it matters is if the glucose comes from inside a muscle cell, if it comes from the breakdown of muscle glycogen, we get 3 ATP. Because when I point out the pathway in the next couple of slides, we get to skip the first step. Okay? We skip the first step. If it comes from glucose that's in your blood, so it's come from glycogen in your liver, it's come from gluconeogenesis done in the liver, or it's come from carbohydrates that you've eaten that are in your blood, you get 2 ATP molecules. Okay? 2. All right. As we said, slower reaction is faster than the aerobic pathways, partly because if we're going to metabolize carbohydrates, this is the thing that feeds the carbohydrates in to aerobic metabolism. So you got to do this first, then you can do the aerobic pathways. Really important thing about glycolysis, okay? Energy production is limited because of something we call end product inhibition. Okay? The end product of the glycolytic pathway. If glucose gets turned into lactic acid, the lactic acid dissociates. You put an acid in water. We told you guys that chemistry would come back at some point. You put an acid in water, which is what the cytoplasm basically is. The hydrogen comes off, so you get lactate and hydrogen ions. That lowers the pH. As you will see when we talk about fatigue, hydrogen ions mess everything up. They inhibit everything. Acidity inhibits everything. Okay? It inhibits glycolysis, it inhibits the Krebs cycle, it inhibits the cross-bridge cycle, it inhibits calcium release from the sarcoplasmic reticulum, everything. Oh yeah, and it makes your muscles burn. So it is very much our enemy in some ways during exercise. Okay? So if you do enough glycolysis and make enough lactic acid, it will turn the system off. Okay? And when the system gets turned off and you can't make enough ATP, then you have to stop exercising. That's kind of the end point of all this. You gotta slow down or you gotta stop. Okay? Okay. What this slide illustrates is here's the blood vessel, there's glucose in the blood, the blue is a muscle fiber. Here's glycogen in that muscle fiber. Okay? If we have glucose in the blood to initiate glycolysis, we bring the glucose molecule in and we stick a phosphate on it. I'll show you guys the enzyme that does that in the next slide. When the glucose comes in, we put a phosphate on it. This phosphate in this first step traps that glucose in the muscle fiber. Okay? You get glucose 6 phosphate. If, okay, we break down muscle glycogen, the glucose comes out of muscle glycogen, it basically already has the phosphate on it. It does not need to be trapped in the cell. It's looking down, the phosphate is there. So you get glucose 6 phosphate coming out of glycogen. Okay? Alright. Okay. We'll do the bottom part here in just a second. Are you guys with me so far? Alright. I'm gonna ask this question and I'm gonna let Doug. Were any of you born in the 90s? One, okay, got one person. Okay. 1, 2, okay, awesome. All right, people. I'm about to show you. I'm about to show you a thing that if it was a TikTok video, we would title it, "It Came from the 90s." Here's a picture from my biochemistry textbook that I still have in my office. I've not set it on fire. Of something that we call the Enden-Meyerhof pathway. Okay. All this is it's every step in glycolysis except for the last one. Okay. It's all of them except for the last one. It is start with glucose, okay, and we end up in pyruvate. And fate, pyruvate controls the fate of all of us. Like with the microphone, you do like that. It's like a Marvel movie, like preview or something. In a world controlled by pyruvate. Okay, so the Enden-Meyerhof pathway is something that I want you guys to know what goes in. Okay, a couple of steps as we go through. It's time to put on a worksheet. We're gonna put some energy in. We're gonna get some energy out. Okay, we're gonna grab some electrons and hold on to them. We're gonna end up with pyruvate. And legitimately, what happens to pyruvate and why is the thing that's the most complicated conceptual thing in metabolism. Okay, so I want you guys to see how we get to the pyruvate before we do anything else. Okay, glucose comes in. Again, this is a 1990s biochemistry book. If you want to see the biochemical structure, it's there. You don't have to memorize it, right? So I know this is you on the test. But you will see in step number 1, the glucose comes in from the blood. There's an enzyme, and I do want you to know this enzyme. It's important, okay? Because it changes dramatically when we do resistance or aerobic training. Time.
That's right. This would be a really good time to get that sheet that I gave y'all out. We're going to cover a lot of those key enzymes and what's produced and made.
There we go.
Sorry. Now, continue.
That's all we've been trying to do us.
Yeah.
Sheets out. The enzyme is called hexokinase. Another one of those things that puts phosphate on there somewhere. Okay? So we take glucose, hexokinase pulls a phosphate off of ATP. So this step takes energy. Okay? It takes energy. We're going to put a phosphate on a glucose and we end up with what's there in step 2 called glucose 6 phosphate. Okay? Glucose 6. That now traps that molecule of glucose in our muscle cells so that we can run it hopefully through the rest of this pathway. Okay, very important first step. If I had glucose 6 phosphate that comes from muscle glycogen, it enters the pathway at step 2, so it misses having to use that ATP molecule to generate it. And that's why we make more energy or we get more energy yield from muscle glycogen. Okay. We then convert the glucose into fructose, right? Tell those people on TikTok fructose ain't so bad, right? We use it in glycolysis. We then are going to use another enzyme that I do want you guys to know because this is going to be what we call the rate limiting step. This is the slowest step in the pathway, okay? And we will talk about kind of the how and why of this probably on Monday of next week. But there's an enzyme called phosphofructokinase. Phosphofructokinase here. We abbreviate it very simply as PFK. And so phosphofructokinase takes another phosphate from ATP, puts it onto our fructose 6 phosphate, and we now have fructose 1,6 bis or biphosphate. somewhere in my biochemistry teacher claps for me. Okay, so far, we've broken this down, we've put two molecules of ATP into this, so we cost ourselves energy to start this pathway. Okay, Amanda? Where does PFK get the phosphate? It gets the phosphate from another molecule of ADP. Okay? So to start glycolysis, there's energy input. Okay, energy input, which I know seems strange, because the point of the pathway is to get energy back out. You gotta give to receive. Okay, so we're gonna put some in, and then we're gonna get more than what we put in back at the end. Okay, anybody remember? You wanna look up here and count carbons, how many carbons are on a glucose molecule? Abby? 6. This is very important, okay? Glucose is a 6-carbon molecule. When we get to the step here, after fructose 1,6-phosphate, we are going to split the glucose in half, or split that 6-carbon molecule in half. And now we have 2 3-carbon molecules, okay? When we get all the way down here to pyruvate has 3 carbons. It's very important. So you start with 1 6-carbon molecule. We're going to get to the middle of the pathway, and we're going to split it into 3-carbon molecules. As we do that here, we're going to do something called a reduction reaction. Okay? As we change the molecular shape, biochemical structure, as we switch between these different molecules, we are going to steal some electrons. Okay? When you pull electrons off of something and hook them onto something else, we call that a reduction reaction. Okay? A reduction reaction. So floating around in your cytoplasm are molecules of something called NAD, NAD plus. Please don't make me, I think it's like nicotinamide, adenine, dyes something, I don't know. We can look it up. Google will tell us. But it's NAD. The point of NAD is to grab electrons and carry them around. So we call it an electron carrier. So we break things down. Boom, boom, we get to here, okay? And for each of our 3-carbon molecules, we're going to pull off electrons and take one NAD molecule, and as we grab the electrons, hydrogen follows. So a proton or a hydrogen molecule will follow. So it goes from being NAD to NADH. Okay? Because there are 2 3-carbon molecules, we will reduce 2 molecules of NAD and NADH. We're going to hang on to these electrons. We're going to do something with them later on. We're just going to set them to the side, and we'll do something with them later. Okay? Okay. You do not have to know what step this happens in. You do not have to know the names of all of these things. All you need to know is it gets split. We pull off some electrons, and as it goes down, we then, for each 3-carbon molecule, we will pull off and make 1 ATP. So at this step, because there's 2, you make 2 here. We get down to here, we make 2 more, okay? And we end up with a molecule, something called pyruvate. So in this process, we started with glucose, we put in 2 molecules of ADP, we generated 2 molecules of NADH, and then we made 4 molecules of ATP and the pyruvate. So if we do the math, 2 ATP in, 4 out, where does that leave us? 2. Upwards of 2. Okay? So we've made 2 molecules of pyruvate, we've netted 2 ATP, and we've netted 2 molecules of NADH. Okay? I want you all, as time put on the sheet, to know what goes in, what comes out, how much ATP we make, that we're getting an ADH, and I want you to know hexokinase and PFK. That's what I want you to know. Okay?
How many net ATP do we produce with muscle glycogen?
Let's talk each other because you just skip the first input. You put in 1, you still make 4, so we have 30. You have 30. Okay. Who has questions? See a lot of talking to our neighbors, right? Is that, going over what you got on your worksheets? You want to take a moment and try and work with your neighbor on the worksheet just for a second. And then if you have a question, you can ask Ty, and I can come around and try to help. We need to get this before we move on, or what happens next is really gonna scramble your brain. Okay? Carla?
Any of the names here,
Correct.
we just have to know that.
So on the worksheet, you're filling in the key enzymes there, and then also those arrows indicate kind of like ATP, ADP,
Right.
and then NAD, NADH. Those are the key players that you need to know from this pathway. That worksheet tells you all of the key players that you need to know. Yes.
So like the open boxes, the first one, it wouldn't be hexokinase, it would be hexokinase.
Yeah, so the blank boxes are gonna be those key enzymes there. And then the arrows are gonna be where you indicate ATP, ADP, and like, yeah, we'll come around real quick, we'll check and make sure that everyone's getting the diagrams correct.
So just to reiterate what Todd said. You're gonna ask yours on the worksheet, okay? You see glucose?
Yes.
The open boxes underneath the glucose are for writing the names of the enzymes at those steps. They're not for writing the structure. The structures you need to know are glucose and pyruvate, which Ty has provided, okay? So those are where the enzymes go. And then the little arrows on the side, Ty, mean ATP.
Yes, so kind of indicate like at each step, right? If we have an enzyme here, we're using energy, right? So ATP goes in, ADP comes out. Similar there, and then down here, those two arrows to the right, I'm just gonna tell you real quick, that's 2 ATP, 2 ATP, right? So we get 4 total, but we get a net of 2. Just kind of indicating, that we're using this energy, getting a yield of 4, but we're having a net of 2 or 3, depending on where we go. And if you pay attention to muscle glycogen, when phosphorylase enters, it's gonna be on that second step, showing you that we don't use that energy again.
You said these are 2 ATP.
Yes.
Are those any?
So those are ATP. That's
Yes.
is where we are in this. So
Like, okay. Thank you. Is it 2? Just right? Yeah.
And then there's something.
Yeah. This is.
Well, yeah, since you know. That's
Better
It's like, it's not in here, it's on the earlier slides. So like the other day, I'm here class, So I told you.
These aren't actually this, we put it, also great, yeah.
Yeah, it's like. Yeah, so like, okay, so these arrows kind of indicate. What are you putting? Sex phosphate, like all the transformations that go through, or is it defined to be? And how long, so that's it. So it goes to the campaign, right? That's what it's showing.
Yeah, basically, all except for use reduce.
Yeah, you don't have the answers, you could. The bigger fighter.
Yes.
Yes.
It was really weird, well, I am. My dad's on so it's yes, I just. Yeah, using that. It's for everyone.
Got away with it. I actually think it's all, it's kind of a new one.
No, it's just two anyway.
Unless you can.
Yeah, it's.
Yeah. But.
So like, see what it's. Same online.
And this is, I don't know how.
And then lots of stuff. Actually, I think it was all real. I'm done with all you. I try not to do it. My friend says you are
Okay. We will solve it, okay? We'll come with what happens to my roommates, and the day will be yours after that, okay? Okay? Have a good weekend, make good choices.
You make the most.
I was once a house. I had choices for me.
Yeah. Maybe even more. With the parents, now that it feels less like. Thank you. Okay, guys.